Thank you, ma'am. I'm delighted to be present at this prestigious conference, and I thank the organizers for giving me this opportunity. I would be introducing you to the concept of cell cycle and a word about the clinical implications of the same. Cell is the basic structural and functional unit of all life on this planet, and hence cell cycle becomes the basic functional process of maintaining life on this planet. Before we introduce you to the concept of cell cycle, it would be better to go through a few basic concepts and terminology related to cell cycle. Cell division is the process by which one cell divides into two, and thus all cells are derived from pre-existing cells. These new cells, as we all know, would be required for the growth of an organism and to replace the cells that are dying or getting damaged or are becoming old. This process of cell division is different in unicellular or prokaryotic organisms and multicellular, more complex eukaryotic organisms. We would be pertaining only to the eukaryotic part. It is imperative during this process of cell division that the daughter cells that are formed from the parent cells are exactly identical in all aspects, except, of course, the size. They are smaller than the parent cell. But otherwise, they have to be identical. For this, all the information is stored in the core control unit, that is the DNA, within the cell. And hence, DNA replication becomes an integral part of cell division. This DNA, before the cell can divide, has to replicate into two copies of the original DNA. And only then will it be possible that the daughter cells be exactly identical to the parent cell. And thus, cell division would result in the parent cell dividing into two exact identical copies of the parent cell, except in terms of the size. Eukaryotic or multisms have all the genetic information stored within structures called chromosomes, and these are 10 to 15 number. Humans have 46 chromosomes, which are arranged in 23 identical pairs. Just a word about the level of chromosomal organization in humans. Human chromosomes, the structure we'll be coming to later in detail. If you enhance or magnify just one part of the chromosome, you'll see that it's made up of a supercoil-like structure. Further enhancement will show that this supercoil again is made up of several coils within. And if you take just one fiber of this supercoil, this fiber is of the DNA double helix, which is tightly bound around proteins called histones. So this is the basic level of chromosomal organization in humans. Now, each chromosome, as I mentioned, is composed of a single tightly coiled DNA molecule, but in a resting cell, a cell that is not dividing, you cannot see these chromosomes individually because these are present in an uncoiled chromatograph. So, in an undividing resting cell, if you see it in a microscope, even an electron microscope, you will not see the individual chromosomes. It is only in the dividing phase that the chromosomes become visible. And these are the histone proteins that I was talking about. A complex of 8 histone molecules forms the histone molecules around which the DNA strand is tightly coiled. This is the basic structure of the DNA. These histones not only provide structural support to the DNA, they also help in functioning. As I mentioned, in dividing cells, the chromosomes are visible in their original form. And so, the structure of the chromosome can be better seen only in a dividing cell. As you can see, the chromosome of a short arm, a long arm, and an identical copy of the same. So, it is formed of two short arms, two long arms, and the center, which is called the centromere. And within these arms lies the DNA along with the histone. A single short and long arm forms a chromatid. So, a single chromosome is made up of two chromatids. The entire set of chromosomes within a cell is the karyotype of an individual, and in humans, as I mentioned, there are 23 pairs. 22 of these are called autosomes, and the last pair forms the sex chromosomes, which determines the sex of the individual. Coming to the actual concept of the cell cycle, as I mentioned, it is the basic functional process that maintains life in an individual. A few facts before we actually move to the cell cycle. How long does it last, a single cell cycle? It will depend on how frequently a cell is multiplying. If a cell is multiplying too frequently, say for example a stem cell, a bone marrow cell, or a cell in a fetus, these cells are rapidly multiplying cells. So the cell cycle is very fast, the turnover is fast, it lasts a few hours. Not just that it lasts a few hours, certain phases of this cell cycle are skipped. As I will be coming to the phases of the cell cycle, there are G1 and G2 phases, and these are absent in cells multiplying, like the fetus cells. However, at the same cells that divide very slowly, or don't divide at all, like the brain cells. Brain cells do it in our entire lifetime. The number of brain cells is fixed in a human body. Then there are cells that divide only when induced by certain stimulant, like the liver cell. If there is injury to a certain part of the liver, the liver increases in size to compensate for that function. So it is induced to increase in size and number. The cell cycle is induced by the injury caused to a certain part of the liver. Similarly, if in the body, then the lymphocytes in the body, which are the immune cells, they reach that place and rapidly multiply in number. So the of cell cycle in lymphocytes is induced by an exogenous. However, there is a Hayflick limit to the number of times a cell can multiply, indefinitely. It is fixed for a particular cell, the number of times it's going to multiply, after which it is going to die. And this limit is called the Hayflick limit. So, the cell can be divided roughly into two stages. This is the M phase or the mitotic phase, the phase in which the cell divides. And the rest, which is marked by the blue arrow here, is the interphase. This interphase is just the phase between two cells. Now, this interphase is further divided into the G of gap 1 phase, G stands for gap, the S or the synthetic phase, and the gap 2 or the G2 phase. These terms were given by Howard and Pelk in 1951 and they still stand. So G1 is, or the gap 1 is the primary growth phase, S is the synthetic phase, G2 is the secondary growth phase of the gap 2, and collectively these form the interphase, followed by the dividing phase, which is the mitosis, in which the nucleus divides into two, and then finally the cytokinesis, in which the cell divides into two. Coming to the gap 1 phase, this phase immediately follows cell division. It is immediately after the cell division. So in this phase, the newly formed cell after the cell division is just maturing. It is maturing to gain its actual function in life, whatever may be the function. The function may be producing hormones, it may be providing protection, it may be combating infection, whatever may be the function. The cell gains its function during this phase and continues its normal metabolic activities. Then comes the synthetic phase. As soon as the cell enters the synthetic phase, it is now starting to prepare for the next cell division. During the gap 1 phase, there was no question of a next cell division. The cell was just maturing. Now it has entered the synthetic phase. So the cell has started to prepare itself for the next cell division. And in this, the DNA, as I mentioned, the most important step before cell division has to occur. DNA has to replicate into two identical copies. This happens in the synthetic phase. As you would interpret from this, before DNA can replicate, it has to be ensured that the DNA that is replicating is normal. And that happens in the gap 1 phase, the G1 phase. It is ensured that the DNA that is entering this phase is normal, that there is no abnormality in the DNA. Because otherwise, the abnormal DNA will be transferred to the offsprings, to the daughter cells. So, a single DNA is divided into two during the S phase. Next comes the G2 phase or the gap 2 phase, and this is the second growth stage. This occurs after the DNA has replicated, and this is the final stage before actual cell division can start. And so, the cell finishes the final preparations for cell division by producing organelles and proteins required for cell division, like centrioles and spindle fibers, which I will be coming to in detail in the mitotic phase. The mitotic phase is the actual division phase. Technically speaking, mitosis is just division of a single nucleus into two. So, division of the cell is not actually a part of mitosis. Mitosis is just the division of the nucleus. And this is followed by the division of the cells, which is called cytokinesis. So, mitosis, also known as karyokinesis, karyo stands for nucleus, is the division of the nucleus into two, occurs only in eukaryotes and has 4 stages. Certain cells do not divide like brain cells. The 4 mitotic stages are Prophase, Metaphase, Anaphase, and Tito Phase. We'll see in all these phases in detail. Prophase is further divided into early and late. Now, as I mentioned, in a non-dividing cells, chromosomes exist in chromatin form. This chromatin in early prophase now starts to condense so that chromosomes become visible now. Also, mitotic spindles start to form during the early prophase. Now, the nucleus is surrounded by a nuclear membrane and it contains an organelle called nucleolus. These are still present in early prophase, but in late prophase, these disappear. The nuclear membrane disappears and the nucleolus is completely broken down. So you cannot see these. The chromosomes are now completely visible. Spindle fibers that will be required later on are starting to form, and they connect the poles of the cell to each other. This is the picture of the late prophase. You can see the nucleus is completely devoid of the nuclear membrane. There's no nuclear membrane around. There is no nucleolus around, and the chromosomes are visible separately. Now, these spindle fibers, this is the single chromosome. These are the long arms and the short arms, and this is the center, which is the centromere. The spindle fibers get attached at the centromere of each chromosome. Now, what are spindle fibers? These spindle fibers are mitotic spindles that are developed from the structures within the cytoskeleton of the cell itself. And they are of the polar type, that is, they run from one pole of the cell to the other pole. These are the polar fibers. Then there are the kinetochore fibers, which run from the pole to the equator, that is the center. All the chromosomes are now lined up at the center or the equator. So the spindle fibers that are running from the pole to these chromosomes are the kinetochore fibers. And there are short aster fibers that are ending blindly from the poles. Now you can see these are the chromosomes that are arranged at the equator. These are the poles, and there are fibers running from pole to pole and from pole to equator. That is pole to these chromosomes, the centromere of these chromosomes. And then these are these small radiating aster fibers. Coming to the metaphase, and just a pictorial depiction of the metaphase, you can see this is an electron microscopic view, and you can see the chromosomes lined up in the center, the poles, the spindle fibers running from pole to pole and from pole to equator, and these are the aster fibers. The anaphase, this is the shortest phase of the cell division. And in this, all that happens is that by the pull of these fibers are running from the pole to the equator. So these fibers pull on the chromatids. This chromosome divides into two chromatids, and each chromatid is then pulled by the fibers to the poles, to the respective poles. So each chromosome is divided into chromatids, and these chromatids are pulled to the respective poles by the pull of these kinetochore fibers. This is all that happens in anaphase. And finally, the Tito phase, where the chromatids reach the opposite poles, the spindle fibers that had formed in order for this process to occur, they completely dissolve. The nuclear membrane again develops. Two nuclear membranes develop at each pole around the chromatids that have collected at each end. The nucleolus, that is one each for each new cell, appears within the nuclear membrane. And then this is where the mitosis ends. Mitosis is technically, as I mentioned, division of the nucleus. Nucleus has divided, two new nuclei have formed. This is where mitosis ends. And this is followed by division of the cell into two. This is cytokinesis. And the chromosomes again disappear into chromatin. So cytokinesis is division of the cytoplasm and an exact division of all the organisms of the cell into two halves, so that each half contains all the organelles. In animal cells, this is you can see this cell division, early cell division. Actually, you can see this under an electron microscope by the formation of a furrow. This is a real picture of a cell dividing, and this is a furrow being formed. This is what we see in an animal cell at the time of cytokinesis. Plant cells slightly differ in that they have a plate. A plate forms for cytokinesis to occur. So the daughter cells that form after this complete cell division process, they have the same number of chromosomes as each other and the same number of chromosomes as the parent cell. They are identical to each other and the parent cells, but smaller. And then they have to mature, as I mentioned. They have to mature in order to gain the entire function for which they are destined. And this happens in the gap 1 phase. Just a brief review of this mitotic process. It is very important. So, eukaryotic cell division is used for growth and repair of damaged tissues. It produces two identical cells. First is the prophase. In the prophase, the nuclear membrane disappears, spindle fibers form, and the nucleolus disappears. This is followed by the metaphase, in which the spindle fibers run from pole to pole, and pole to equator, and the chromosomes are arranged at the equator. This is followed by the anaphase, in which the chromosomes, or actually the chromatids, are being pulled to their respective poles. This is followed by, finally, the Tito phase, in which the two cells are divided, nuclear membrane appears at each end, nucleolus appears at each end, and the mitotic spindles disappear. And the two cells finally divide in cytokinesis. Animated picture of the same. The chromosomes are becoming visible. They are accumulating at this equator. Mitotic spindles are formed, and they are pulled to the poles. Nuclear membrane forms, and finally cytokinesis occurs. This is the entire process of cell division. Now, as I mentioned, cell cycle is the basic functional process of all living organisms. So what happens when this cell cycle becomes defective? If the cell cycle becomes defective, then the cell will cease to function in its normal manner. Either it will behave in an abnormal manner, it will function abnormally, or if there is a loss of control of the cell cycle, then the cell will continue to multiply indefinitely. And that, in a sense, is the basic concept of carcinogenesis. A cancer develops because of loss of this control of cell cycle, and hence the paramount importance of controlling the cell cycle. So there are 3 points where nature has given 3 points within this cell cycle in order to control any defects that occur during the cell cycle. First is the G1 checkpoint, which occurs just before the cell enters the S phase. Then there is a G2M checkpoint between the G2 and the M phases. And finally, there is a lesser important M checkpoint within the mitotic phase. Now, these checkpoints are made by nature in order to ensure that any exogenous or endogenous carcinogen could be tobacco, alcohol, lifestyle, anything that produces a mutation within the DNA, a defect within the DNA, that will ensure that the cell cycle goes on indefinitely, and hence a tumor forms. Nature has given these checkpoints to ensure that such a DNA is not allowed to replicate. Such a cell which is defective is not allowed to proceed. However, as we all know, still happens, and hence cancers develop. Now, there are special proteins and enzymes within the cell that control this cell cycle. And as I mentioned, this is a G1 checkpoint which prevents entry into the S phase if the DNA is defective, and the G2 checkpoint which determines which cell is going to enter the mitotic phase. And finally, the M checkpoint, which within the mitotic process decides whether to terminate the mitotic process there or whether to continue. Simply speaking, there are 2 aspects of control of any process, any natural or artificial process. One is the activator part and the inhibitor part. It is a balance between these 2 arms that determines which way the cell cycle is going to proceed. If there's too much of activation, the cell cycle is going to go on. Too much of inhibition, it's going to cease. Activation is done by something known as cyclins. Cyclins are simply proteins within the cell, and they associate with their respective enzymes. These enzymes are called cyclin-dependent kinases, or CDKs. So, individually these 2 are not active, but when they combine together, cyclins, when they unite with their respective CDKs, they become active, and together they can activate the cell cycle. Inhibitors, there are a number of inhibitors, but 2 important indigenous inhibitors within the cell are the Retinoblastoma Protein and the P53 proteins. These are very essential to inhibiting the cell cycle and hence for its control. And hence these are frequently mutated in human cancers. Cyclins, as I mentioned, are present within the cells, but it is the level within the cell that determines whether the cyclin is going to act or not. So whenever a cyclin is needed, it increases in level and combines with the CDK to act on certain targets. So the cyclin combines with its respective CDK, and together then they become active. These are various types, we don't have to go into the details of that. Once these are active, they cause activation of the cell cycle. And how do they act? For that, first we have to understand what retinoblastoma protein does. Now, retinoblastoma protein is sitting at the G1S checkpoint. What does it do? In the resting state, it is active. It prevents the cell from entering the S phase. So long as the retinoblastoma protein is acting, the cell will not enter the synthetic phase. The cell cycle will not proceed, the cell will not divide. Cancer will not occur. So, whenever the cell cycle has to proceed, cyclin and CDK, they come and they phosphorylate the retinoblastoma protein. The retinoblastoma protein becomes active, this inhibition is lost, and the cell cycle can proceed. So, in a cancerous condition, either the retinoblastoma protein is defective, so this inhibition is lost, the cell goes on multiplying, or the cyclins are too active, so the retinoblastoma protein, although it's normal, cannot function. Next is the P53, which is another protector of the cell against development of cancer. So what does this P53 do? It has a supportive role to the retinoblastoma function. P53 has the capacity to check whether there is any mutation within the DNA present in the cell. So if there is any mutation within the cell, P53 will not allow the cell to further progress into the S phase till that mutation is checked, repaired by the reparative enzymes. If it cannot be repaired, then P53 will ensure that the cell dies rather than proceeding to produce two identical daughter cells which will have the same defect. So P53 will cause cell death if there is a defect in the DNA which cannot be repaired. And how does it act? As I mentioned, cyclins inactivate the retinoblastoma proteins. So P53 inactivates the cyclin CDK complex. So as soon as the cyclin CDK complex is inactivated, the retinoblastoma protein is free to act and it halts the cell cycle. The cell cycle cannot proceed. So P53 has a supportive role to the retinoblastoma function. So in a nutshell, activation of growth is by cyclins and CDKs, and if there is too much activation, the cells will continue to increase in number. Normally this might be required in a normal human being also, but that is in a controlled manner. If it is in an uncontrolled manner, it forms a tumor. Inhibition of growth is by retinoblastoma and P53, causes decrease in cell number, or apoptosis. Apoptosis is nothing but programmed cell death. A cell is destined to die sometime during its life, and that is by the process of apoptosis. Sometimes this is also pathogenic, decrease in cell number, like a cardiac failure may occur because of excessive decrease in cell number. The cells continue to die, so a cardiac failure may occur. But from the tumor or the cancer point of view, it is the activation part which we are more concerned with. Too much activation, uncontrolled cell division, cancer. So this is just the same as I mentioned. And the third is the spindle assembly checkpoint, which is within the mitotic process. And if you remember the Metaphase and the Anaphase, it is between them. And it can be still arrested at that point. The cell division can be arrested at that point if the cell is defective. It is of less importance in humans. So how is cell growth regulated? It is very tightly regulated in a normal individual. And this is by various processes like contact inhibition. Too many cells in a small space, then they inhibit cell division in neighboring cells. This is contact inhibition. Each cell has a finite number of cell divisions, after which it dies, the Hayflick limit. Then there are genes within the cells which ensure that there is a control on the cell cycle. There are Proto-Oncogenes. Proto-Oncogenes means genes that have the capacity to become into cancerous genes. And these, as the name would suggest, they stimulate growth within the cell. And if there is a mutation in these genes, these proto-oncogenes become oncogenes, and then they produce uncontrolled cell growth and cancer formation. So these proto-oncogenes could either be growth factors, they could be receptors on the cell which promote growth, these could be CDK cyclin-dependent kinases, or these could be factors which enhance protein production. Tumor suppressor genes, on the other hand, whenever they are active, they will inhibit cell growth, like P53, retinoblastoma. These are tumor suppressor genes. And if there is a mutation in these genes, then cell growth will be uncontrolled and hence cancer formation. A very common example is the BRCA1 tumor suppressor gene. This is a common tumor suppressor gene that is mutated in inherited breast cancers. Similarly, P53, it is mutated in almost half of human cancers. Half of human cancers will have some form of P53 mutation. And whatever happens to these destroyed cells, they undergo apoptosis, which is, as I mentioned, programmed cell death. And this is genetically regulated. Just a brief word about how this happens. A mutation in a P53 gene. Now there are 2 copies of this P53 gene. So if there is a single mutation, cancer will still not develop. But this person who has a single mutation will be at a predilection for cancer formation because all he needs is another mutation. And this is environmentally induced, like by tobacco or alcohol. All these can cause mutations within the DNA. And he needs just a single mutation as compared to a normal individual who needs 2 mutations. So he is predisposed to cancer. And what can cause this damage? As I mentioned, radiation, toxins, which is tobacco, alcohol, the 2 common toxins, chemicals, and hence the importance of changing this environmental procedure, environmental carcinogen exposure by avoiding sun exposure, smoking, alcohol, diet restrictions in amount and content, increase fiber, decrease salts, and most importantly, early detection, particularly in patients who have a family history of cancer. Just a brief word about apoptosis. Apoptosis is a process of programmed cell death, and any cell which is not allowed to progress in the cell cycle undergoes apoptosis. This is what we want as an oncologist. We want cells to die. Cells, when they are replicating in an uncontrolled manner, that is when the tumor develops. Within the tumor, we want apoptosis to occur. So apoptosis is an oncologist's friend. The various definitions, it is derived from apo, which is separation from, and tosis, which is dropping. And the importance of apoptosis lies in the fact that, as mentioned, first is tumors that develop when the failure of cells to die occurs. That is, this apoptotic mechanism is lost. That is when tumors develop. And the immune system tolerance can occur because of loss of apoptosis. On the other hand, inappropriate apoptosis can cause excessive cell death and things like cardiac failure. And it is important in the process of morphogenesis in embryonic life. From the purview of cancer, there is intrinsic inhibition of apoptosis in cancer cells. And it has been proved that the various targets in this apoptotic process which we can use in order to kill these cancer cells. There is various mutations within the cancer cells which do not allow them to die. And these targets can be used to produce drugs that act on these targets and cause death within these tumors. A number of these targets are under investigation. Drugs have been developed. They are under trial. Trail receptor, BCL2, IAP family, Survivin. There's actually a poster outside on Survivin. Finally, to summarize, cell cycle is the basic functional process of all cells. It is of paramount importance in the homeostasis of a living being. Cells have to function normally for a person to function normally. Cells to function normally, cell cycle has to proceed normally. And hence, it is a tightly regulated process. There are activators, CDKs and cyclins, which together activate the cell cycle. Too much activation leads to uncontrolled cell division and hence tumor formation. Too much inhibition leads to excessive decrease in number, loss of function. And defects in this results in various diseases and from our purview, cancer. And it is the study of these defects that is the basis of all research in carcinogenesis. The entire research in carcinogenesis is to elucidate these defects, to determine what is the target of these defects, what is the cause, and what can we do, what drugs we can give to compensate for these defects, and hence these form important and very important targets for therapy. I hope I have been able to introduce you to the concept of cell cycle without being too technical, and again thank you for giving me this opportunity. Thank you.